Yeah, in the previous lecture, we had discussed the general structure of the antibody and had said that it's the common structure except that the CH2 domains and the hinge region is different. The CH2 domains are extra, that is, it is up to CH4 in IgM and IgE. Otherwise, the rest of the other three antibodies, that is IgG, IgA, and IgD, have only three CH3 domains. And I had also mentioned that the constant region domain is the one that is used for classification of the antibody. And if it is gamma, so it is labeled as gamma 1, C gamma 1, C gamma 2, C gamma 3 for the gamma globulin, that is IgG. As shown in this diagram, you can make out that it is labeled as C gamma 1, C gamma 2, gamma 3. Okay. So this is the highly simplified structure of an antibody with the hinge region showing two disulfide bonds. Okay. And the variable region is labeled as VL and CL. Constant region as C, gamma 1, gamma 2, gamma 3, and variable region of the heavy chain is labeled as VH. Okay. And I had mentioned briefly that IgG can be classified into four different classes. Okay, four different classes as IgG1 and G2, G3 as represented here in this slide in the diagram. Okay, before that, the major antibody that is in circulation in any individual to protect from the infection is always IgG. And it is the longest living antibody in the body. It will live for a long time, up to about 23 days. Once it is produced, it can circulate up to 23 days. And once it has formed the complex with the antigen, it is going to be destroyed by the complement mediated lysis because it is going to be taken up by the macrophages because of formation of the complex with it with C3b complement, and through the C3b receptor or FC receptor of this antibody, it is going to be engulfed together with antigen which has been formed a complex with it. Then it will be destroyed. Then it will not be there for 23 days. But the unbound antibody which is produced against an antigen will be in circulation for 23 days. Okay. Now these different forms of IgG are classified based on their difference in amino acid sequence in the constant region and plus the number of disulfide bonds in the hinge region. The typical antibody, I had said that it has two disulfide bonds in the hinge region, and IgG1 has two disulfide bonds in the hinge region, whereas G2 and G4 has four disulfide bonds in the hinge region. But the positioning of these disulfide bonds, even though in this diagram is looking alike between G2 and G4, it is not the same. Whereas in G3, you can make out that longest hinge region is there with 11 disulfide bonds. So that itself proves that these antibodies differ from one another, even though they belong to the same class by few amino acid residues in the constant region. Plus other constant region domains, that is CH2, that is C gamma 2 and C gamma 3 domain, also have difference in amino acid sequence. Therefore, the subtypes of immunoglobulins in a particular class, the subtype in a particular class are called as allotypes. Allotypes. I will give the detailed explanation about the allotypes in the later class. Okay, for the time being, you remember subclasses of immunoglobulins under one major class are called as allotypes. Now, what is the role of these different types of IgG molecules? All of them are meant to tackle the antigen, but they have a different role to play. And they vary in concentration in the serum of a healthy individual. For example, the molecular weight is not going to be different, of course. Molecular weight will remain constant, that is about 150,000 only, the molecular weight. And their electrophoretic mobility also is very poor electrophoretic mobility. They move a very short distance when you carry out electrophoresis between pH 7.5 to 8 at that pH. Okay, and they don't move far away from the pole, negative pole. They move very short distance and they be remaining there. Therefore, they are called as very poor electrophoretic mobility molecules. Okay. So IgG1 is about 9 milligram per ml in the serum of a healthy patient. IgG2 is 3 milligram per ml and IgG3 is about 1 milligram per ml and IgG4 about 0.5 milligram per ml. They differ in concentration, but 75% of the immunoglobulin in the body is IgG class. Okay, it is all these subclass put together, it is about 75%. And what is the role of these different IgG class of immunoglobulin? Let us see. So IgG1 and G3 and G4. Okay, IgG1, G3 and G4. These three different molecules which are there, allotypes, can easily pass through the placenta. That means they can diffuse from the mother's placenta into the fetus. So that means they play a major role in fetal circulation. They are the antibodies that give protection to the fetus by being in fetal circulation, by diffusing through the placenta, and they are produced by the mother, and they give the protection to the fetus, developing fetus in the mother's womb. That is IgG1, G3 and G4. That means they have the receptor on their Fc arm to bind to the placental region and get transported across the placental region of the mother to the fetus. Then the IgG3 has the special ability of binding to the complement. But remember, IgG class of antibodies are not complement fixers. There is a necessity always for two molecules of IgG to come come and associate side by side at a distance of 100 angstroms so that the complement receptor can be bound to the CH2 domain of this antibody and then initiate the complement mediated lysis or whatever it may be. Even then, the IgG3 is the best complement fixer, means complement can bind to the CH2 domain of IgG3, followed by IgG1, then IgG2, and the least means there is no binding ability for IgG4. Complement will not bind to IgG4 because it doesn't have the receptor for the complement. So highest is IgG3 followed by G1, then G2. So even then, they are capable of bringing about complement mediated lysis because the two molecules of them should be associating at that particular region to get rid of the antigen antibody complex. Then IgG1 and G3 are capable of binding to the Fc receptor. And when I say Fc receptor, I kept mentioning that some of the immune cells like macrophages, dendritic cells, then the natural killer cells, then eosinophils, neutrophils do have Fc receptor. And when it's specific to the gamma globulin, that Fc receptor is called as Fc gamma receptor. It is meant for IgG binding. That Fc region of the antibody goes and binds to that receptor on the membrane surface, and therefore it initiates certain signal transduction processes because it has got, once it goes and binds there, it has got the cytoplasmic Fc receptor domain has got about 25 to 26 amino acids. It can bring about signal transduction. Okay, and by binding to the Fc receptors, they facilitate the uptake of the antigen that is complexed with their Fab arm through endocytic process or phagocytic process. And that is why they facilitate the destruction of the antigen. And that antigen could be a soluble antigen. And that soluble antigen, when it has formed a complex with IgG, that complex is going to give rise to preceptin, and the preceptin is nothing but the precipitate that has accumulated because of the antigen antibody reaction, and that precipitate is going to be removed through phagocytic process or endocytic process by the macrophages, which are scavenger cells. through the Fc receptor. So that is what is facilitated by IgG1 and G3. Okay, so immunoglobulin G is meant for precipitin reaction for the soluble antigens. In addition, it is also capable of binding to the bacteria and viruses. It can be synthesized against bacteria and viruses when they enter, and these become particulate antigens. And when it binds to these particulate antigens, it can facilitate their uptake. Okay, and I'd use the one of the word called as opsonization. When the antibody binds to the membrane of the bacteria, that membrane of the bacteria is going to become distorted, as well as it will be denatured, and opsonization takes place, and it facilitates uptake of that bacteria by the phagocytes and the destruction of that bacteria. In addition, the viral particles, when they bind to the FABR of the antibody, that also facilitates viral uptake by the macrophages and destruction of the macrophages, as well as by the neutrophils. So therefore, it is also called as bactericidal and virucidal antibody. IgG antibody is also named as bactericidal virucidal antibody, and it is also called as precipitin antibody. Why? Because major antibody involved in soluble antigen precipitation is IgG. So precipitin antibody. These are the alternate terminologies that have been used for the IgG antibody. And I had mentioned that it has the tremendous ability to pass through the blood vessels. It can diffuse out of the blood vessels, means it can be made available immediately. And let us say, for example, there is IgG in circulation, and somewhere the wound takes place, and the same antigen has made its appearance, and IgG was produced against it long time back. The immune boosting takes place, immune memory cells will respond, and IgG will be produced within 2 days time, within 48 hours, and that will start diffusing out of the blood vessel to tackle that antigen where the wound has taken place. Diffusion through the blood vessel, diffusion through the placenta is only possible for IgG molecule. Okay, so it was the first one to reach the fetus, first one to reach the site of infection by passing through the blood vessels. That is the tremendous ability of IgG. Only thing, the disadvantage of IgG is that it is a poor agglutinating antibody. Poor. It cannot agglutinate the particulate antigen so efficiently as IgM does. Okay, we will come to that little later. IgG is a poor agglutinating antibody, poor complement fixer. Therefore, large amount of IgG will be required to initiate the complement mediated lysis as well as agglutination reaction. Okay, and if at all it fails to cause agglutination reaction, remember I had explained in Coombs test, Rh antibody is IgG antibody. Therefore, it does not cause a very strong agglutination of Rh positive RBCs. To bring about strong agglutination, you need to bring about a secondary antibody which is produced against this IgG, which forms a bond with this IgG, that is anti-antibody, and the secondary antibody is produced in the animal, and you cannot produce secondary antibody in human beings. So that animal antibody can cross-link this IgG molecule and bring about agglutination. Then the agglutination becomes stronger because it is already bound by the antibody, that is Rh antibody, the Rh positive RBC. To that you are cross-linking with the anti-antibody, that is anti-Rh antibody. Okay, so that is what I meant by poor agglutinating ability. Okay, so the IgG, which is capable of bringing about agglutination, is weak agglutination, but strong you can bring about using the anti-antibody. Okay, so IgG antibody is a major antibody that gives protection, and I had mentioned that It has got a carbohydrate moiety or carbohydrate residue in the CH2 domain, which is labeled as CHO. And that carbohydrate moiety gives the hygroscopicity for the Fc arm of the IgG, and the percentage of carbohydrate is about 3% here. That is more than sufficient. 3% of carbohydrate, and it is a heteropolysaccharide made up of glucose, galactose, and mannose polymers, which are bound here, which facilitates the binding of water molecule and hygroscopicity of the IgG molecule. And therefore, it is easily soluble in water. It is soluble in water. Okay, so that is as far as IgG molecule and its allotypes are concerned. Now let us move to IgM molecule. Look at the structure of IgM molecule. IgM is a pentamer. Now each of these are monomers of IgM, and 5 monomers are bound together by disulfide linkage to form a pentamer. Each monomer of IgM has an FAB site, 2 FAB sites. That means a pentamer IgM molecule can bind to 10 antigens at a given time. All right. If it is a large molecular weight antigen, both these FAB sites can be occupied by one antigen. Therefore, it can bind to 5 antigens at a time. And because of its binding ability to 5 antigens or 10 antigens, it becomes a strong agglutinating antibody. Therefore, IgM is the antibody that is produced against our blood groups. Anti-A antibody is IgM. Anti-B antibody is IgM. And these antibodies can agglutinate the blood easily, because they have 10 antigen binding sites. Okay, and remember, I had mentioned that the IgM molecule has got 4 constant region domains, which are labeled as CMU1, CMU2, CMU3, CMU4. Okay, and there is only one disulfide bond holding the heavy chain together. Only one disulfide bond instead of 2 disulfide bonds, and there is no hinge region. And that disulfide bond is present between the CH2 domain and CH3 domain, or CMU2 and CMU3 domain. One single disulfide bond is there, which attaches or which holds the 2 heavy chains of the antibody. Then the cross-linking of each monomer, is brought about by disulfide bonds between CMU3 and CMU4 or CH3 and CH4 domains, okay, of each monomer are cross-linked to bring a pentameric structure. Pentameric structure is constructed because of the intramolecular disulfide bond plus intermolecular disulfide bond. The intermolecular disulfide bonds are between CH3 and CH3 of the other monomer, CH4 and CH4 of the another. Okay, so there is one J chain. What is this J chain? J chain is the joining chain. As the IgM molecule is going to be produced by the B lymphocyte, all right, it will be appearing as monomer. Okay, it is not synthesized as a pentamer. It will come out of the B lymphocyte, secreted out of the B lymphocyte as a monomer. As soon as 2 monomers make their appearance, all right, 2 monomers, they are joined by a J chain, J for joining, which is a small peptide of 15,000 molecular weight with a disulfide bond to form a loop within itself. It forms a hairpin loop because of the disulfide bond, 2 disulfide bonds within itself, intra-chain disulfide bond. And then from the NH2 domain as well as COH domain, 2 cysteine residues that are present on either side, one in NH2 domain, another in the COH domain, establish a disulfide bond with the 2 monomers at CMU4 or CH4. Okay, the CH4 domain of the two monomers are arrested by the J chain. Now you have first dimer formed by the J chain. As soon as IgM is secreted by the B lymphocyte or plasma cells, the dimers are synthesized, or dimers are created by the J chain, and this J chain is secreted by the epithelial cells as well as the endothelial cells of the blood vessels and the epithelial cells found in the lymphatic channels. Okay, so joining chain or J chain of 15,000 molecular weight first facilitates the construction of a dimer. Once the dimer is created, that dimer will establish a first disulfide bond between the two dimers through CMU3 linkage. Okay, and it is already linked to one another. That is, in fact, between the heavy chains, there is already a single disulfide bond. Then the other three molecules will start joining that dimer through the disulfide bonds of CMU3, and CMU4. That is how the pentamer is constructed. And once the pentamer is formed, that pentamer is in circulation of the human blood for 5 days. And the first antibody to make its presence in the blood of an individual against any antigen, first antibody is IgM. Why? Because I had mentioned yesterday or day before yesterday when I had dealt with the B lymphocytes that the first receptor on the B lymphocyte that is going to be put up on its surface is MIgM, monomer of IgM. And that monomer will be exact replica of one of the monomer of these pentamer. And all these monomers are identical to each other. Means they have undergone clonal selection against that antigen. They have undergone affinity maturation, means strong affinity binding ability they have established by permutation and combination of the genes of the B lymphocyte, which have put in a best receptor. And that makes this IgM molecule the best molecule for that particular antigen. So when it is in circulation, when it is constructed into a pentamer, it is there in the blood for 5 days. It is a short-lived antibody. First formed antibody and short-lived antibody. And most efficient agglutinator. And because these molecules, monomers of the IgM, are placed close to each other and they are within that requirement of 100 angstroms. Since they are placed close to each other already in the pentamer, they facilitate binding of the complement readily here. That C1q domain of the complement comes and binds readily here when the antibody is bound by the antigen in the FAB region. And complement gets activated immediately. Therefore, IgM is considered to be a strong complement activator compared to IgG. A single molecule itself will be sufficient to activate the complement and bring about complement mediated lysis. Let us imagine that among these monomers, any one of the monomer will establish the contact with the bacteria, bacteria being a particulate antigen. It can initiate the complement mediated lysis immediately after it binds to the bacteria. And it can efficiently bind to 5 different bacteria or 10 different bacteria depending on the size of the bacteria as well as the viral particles. Okay, only thing is it doesn't facilitate phagocytosis because being a large macromolecule, none of the cells have Fc receptor mu for on their surface for phagocytosis because it is being a macromolecule. So individual molecule if I take, individual monomer if I take, it is about 180,000 in molecular weight. And when all pentamers are put together, it will amount to about 900,000 molecular weight. Okay, Each monomer is 180,000 and the pentamer is 900,000. Therefore, because it assumes a large shape, it is called as macroglobulin. IgM got the name M because of the mu protein chain, plus it is a macroglobulin, large globulin molecule of large molecular weight. Okay, once again, it shows very poor electrophoretic mobility. Wherever you load it in the electrophoresis during the gel electrophoresis, very short distance it will travel. Because of the molecular weight, it can't travel large distance. Okay, and the concentration of this antibody is found to be about 1.5 milligram per ml in the blood, but it is not there always because it is short-lived. As soon as the infection takes place, and this is one of the antibody that is detected for coronavirus, and within the 5 days after the infection of a person, 5 days and up to 7 days, 5 to 7 days, you can detect this IgM antibody in the blood serum of a person who is infected, and after that it will disappear. It is going to be taken over by the IgG, and that class switching mechanism I've already explained briefly. Class switching takes place and IgG will take over, and then you will see that after 7th day onwards up to the month and 1 month, the IgG will be there in large quantity. Therefore, for the detection in a patient, both IgG and IgM are detected in some patients. Suppose if you carry out the detection test after the 7th day, you can get IgG. But if you do it before the 7th day, you can get only IgM. Okay, IgM against the virus. All right. So this macromolecule or macroglobulin is present in about 5 to 10% in the blood. 75% is IgG, 5 to 10% is this molecule IgM. First few days, that is 5 days of the infection, it will be there, short-lived antibody. Okay, and I have already told that it is a strongest complement fixing antibody, bring about complement mediated lysis. All right, and then most important feature of the IgM molecule is that because of its pentameric nature and being macromolecular nature, it cannot diffuse out of the blood vessel. It is intravascular. It cannot diffuse out of the blood vessel. It cannot diffuse through the placenta. Okay, but it can make its presence in the mucosal lining of the individual, just like the way in the mucosal lining you may see IgA antibody. The next antibody that you may see in the mucosal lining is IgM. But once again, it is short-lived. In the mucosal lining also, it will be there for 5 days for defending. After that, it will disappear. Right? And I've told you, because of its macromolecular nature, it cannot attach to the Fc receptors and doesn't have Fc receptors. Except that only the monomer, membrane-bound IgM monomer, has the receptor on the B lymphocyte and becomes the major receptor of the B lymphocyte. Okay, B cell receptor, BCR in short form, constituted of a monomer of the IgM. And the same monomer replica of high affinity will construct a pentamer, and that pentamer will be there in circulation of the activated B lymphocyte. Therefore, in circulation, in the blood, you will not be able to detect the monomer. If you see the monomer, it is attached only to the B lymphocyte. No other immune cells have the receptor for monomeric IgM. Okay, so therefore, the IgM is a predominant antibody that can tackle bacterial and viral infection initially. Whenever there is initially high dose or high amount of bacterial infection or high amount of viral infection is there. When the antigen concentration of these two particulate antigens are very high in the blood, the IgM molecule tackles it. It can easily bind through its 10 FAB sites to these particulate antigens and bring about the destruction of these particulate antigens by initiating complement mediated lysis. Okay, complement mediated lysis. Then the carbohydrate moiety or carbohydrate residue compared to IgG molecule is very high, right? It is very high. The carbohydrate moiety in the IgG molecule, IgM molecule is very high. This is the simplest diagrammatic structure of IgM, and that carbohydrate moiety is bound from C1, that is CH1 or C mu1, CH2, CH3, CH4 domain. It is all across all the domains, whereas in IgG molecule, I had said that it is bound only to the CH2 domain. Okay, therefore this generalized structure in that CHO is shown only in the CH2 domain. CHO stands for the carbohydrate, and it is about 3%, but here it is about 12%. 12% of carbohydrate is there, which is bound to the C mu1, C mu2, C mu3, C mu4 domain. And the type of carbohydrates that are bound are different here. C mu1, mu2, and mu3, okay, C mu1 and mu2, mu3 are bound by actually branched carbohydrate moieties. Okay, branched carbohydrate moieties. And those branched carbohydrate moieties constitute the important sugars, which are going to be the monosaccharides, which are polymerized to form heteropolysaccharide, and those will be fucose, mannose, galactose, N-acetyl glucosamine, and once in a while the sialic acid. Okay, sialic acid. So these are in polymeric form, bound, branched in C mu1, C mu2, and C mu3 region. And unbranched residue, two of them, unbranched residue, two of them each, okay, bound at the C4 domain, okay, C4 domain as two different chains as mannose and N-acetyl glucosamine. They are unbranched. The polymer of mannose and N-acetyl glucosamine, the two chains of that as polymers, heteropolymers, are bound to the C4 domain. Okay, and by binding in such large quantities to the constant region domain of the IgM, the carbohydrates facilitate hygroscopicity, increased hygroscopicity of IgM molecule. Even though it is a macromolecule, it is soluble in water. Okay, it forms a homogeneous, nice suspension, and therefore it can be easily used. Okay, there is no need for the production of separate IgM. One has to give an injection into a human being of the opposite blood group, a small dose of opposite blood group injection. And when you give to a person, B blood group RBCs, a small dose of that person will produce large quantity of anti-B antibody, which is of IgM type. And since it will be intravascular, when you bleed that person and take out some blood from him, large quantity of antibody can be harvested. And that is the antibody that is available in the diagnostic kits. In the diagnostic kits that are available in the laboratory, when you go for blood group checking, that is the antibody that is added with, actually it is preserved by adding some preservative, and that is added to your blood to cause agglutination. Okay, so remember that the IgM antibody, which is intravascular, meant for tackling the antigens that enter your blood vessels, blood passages, including the lymphatic vessels. Whatever the antigens that are there which have invaded the blood vessel, they are tackled by the IgM antibody until IgG takes over, until the class switching mechanism happens. And being intravascular, it can give you protection in the blood vessels. Okay, and first few days of the life of a young one, young born, whenever the child is born, it is thrown open to the environment. Until then, it was protected in the mother's womb. In the mother's womb, it was protected from the environment. It was not in contact with any bacteria or virus that is found in the environment. And it was gaining the immunity, or passively it is being protected by the mother's antibodies. And those mother's antibodies which were going into the fetus all of them were of IgG class, because they can pass through the placenta. And first, that protection to the fetus passively was happening in the mother's womb. As soon as the fetus is born, it is thrown into the environment. It will start respiring. Until then, it was not respiring through its lungs. All the oxygen it was receiving was through the diffusion of the mother's blood oxygen through the placenta. Okay. Now, once it starts breathing, first antigen entry takes place through the nasal surface. And once it starts feeding on the mother's milk, first antigen entry will start taking place through the oral route. Remember that. So the fetus will be exposed to now bacteria and viruses that are there in the environment. But passively, it had gained the antibodies from the mother. Those antibodies will give the protection to the fetus, which is there in circulation in the fetus even after its birth. Then, if mother starts feeding the milk, through the milk, IgA will be transported to the fetus, into its gut. And that IgA will play a major role in giving protection to the fetus. And that is what I'm going to talk about in the next class, that is when we meet on 19th, that IgA as an immunoglobulin for the fetal protection. And I told you that IgA is not going to be digested by the fetal digestive system. Okay, so whatever it may be, then the fetal bone marrow will be stimulated because the fetus, after its birth, it is exposed to the environmental antigens, the bone marrow will be stimulated. And that bone marrow will start responding now. And the fetal thymus, the child's thymus, as well as bone marrow will be highly, highly active to initiate the immune response. But remember, first to 6 to 8 months of the fetal or the child's protection is wholly dependent on the mother. If the mother feeds the milk, the child is protected. If the mother does not feed the milk, and if she doesn't have the milk, child needs to be protected passively by giving some antibodies to the fetus, then only the fetus or the child can survive. Otherwise, it is very difficult for the child to fight the infection that is going to be caused by the environmental pathogens, because the passive transfer of the immunoglobulins that has taken place in the child will not last long. All right, the immunity will take for the child to develop on its own about 8 months. After the 8 months, the child will start producing its own B lymphocytes, its own T lymphocytes, and its own cell-mediated immune response and humoral immune response. Until then, the child has to be protected by the mother's milk, and that is through IgA. And that IgA is a very important molecule. Okay, so remember, the IgM molecule will not exist as a monomer in the serum. It will exist as a pentamer, and monomer is seen only bound to the B lymphocyte as the receptor of the B lymphocyte. Okay, thank you.